Assalamu alaikum. I am Nimra Farooq and today we will solve the questions at the end of microbiology book from the section of basic virology. So let's get started. Our first question is viruses enter cells by absorbing to specific sites on the outer membrane of cells. Each of the following statements regarding this event is correct except in this question they tell us that how viruses enter into the cell and we have to identify which statement is not correct about this phenomena. Option A, the interaction determines the specific target organs for infection. Viruses are highly sensitive. Their binding to specific receptor tell us the target organ which they want to affect or infect. So this statement is correct. Option B, the interaction determines whether the purified genome of a virus is infectious. Purified genome is something when scientists want to study a specific part of the DNA or RNA, they remove any unnecessary material from that piece of DNA or RNA. So that part will be marked as purified. And of course, DNA or RNA, it alone cannot cause the disease. It must have to have the remaining part of a virus such as capsid, envelope or whatever it has to produce disease. Alone, a purified DNA or RNA is not able to cause disease. So this option is not correct. Option C, the interaction can be prevented by neutralizing antibody. As we know that virus only bind to the receptors, and of course if the receptor is occupied by something else, any antibody or whatever it is, then where the virus will bind. So this statement is also correct. Option D, if the sites are occupied, interference with the virus infection occur. Again that receptor thing, if the receptor get interacted with virus, of course the virus produce disease or infection. So answer is option B, which is incorrect and all other options are correct. MCQ number 160. Many viruses mature by budding through the outer membrane of the host cell. Each of the following statements regarding these viruses is correct except in this MCQ you should have the knowledge about budding. You know what is budding? When a virus get into the cell, it releases DNA or RNA, whatever it has. It infects the genetic material of the host cell, and after infecting, it want to get out of the cell. For that purpose, it pushes the membrane of the infected cell so that it can go out. In this struggle, a bulge is formed in the outer membrane, and when the virus came out, a envelope of the host membrane cell is surrounded around that virus. This process is called budding. Now keep this concept in your mind to solve this MCQ. Option A, some of these viruses cause multinucleated giant cell formation. You know what is multinucleated giant cell? When the virus cause infection in one cell, it attracts the neighboring cell to come closer. When the neighboring cell come closer to the infected cell, they all get together and make a giant cell. This giant cell is called multinucleated giant cell because it has nucleus of many cells. So option A, that some of these viruses cause multinucleated giant cell formation. Yes, they do. Option B, some new viral antigens appear on the surface of the host cell. Of course, when a virus alters the cell genetic material, the cell displays some changes on its surface. These changes are called antigen. So this is also a correct statement. Option C, some of these viruses contain host cell lipid. I told you earlier that in budding, when the virus pushes himself out of the cell, it gets surrounded by the envelope made up of host cell membrane, and the cell membrane is made of lipid. So this is also a correct option. Option D, some of these viruses do not have an envelope. This is wrong statement. Again, the concept of budding. If the virus undergoes budding, it must have an envelope. It is not possible for the virus to get out of the cell without that envelope. Question number 161. Biochemical analysis of a virus reveals the genome to be composed of 8 unequally sized pieces of single-stranded RNA, each of which is complementary to viral mRNA in infected cell. Which of the following statement is unlikely to be correct? In this statement, you only have to remember purified. We discussed it in the previous MCQ. They are saying that when the messenger RNA is purified from the infected cell, they reveal that it is composed of 8 pieces. Option A. Different proteins are encoded by each segment of the viral genome. Do you remember codon? Each has different instructions. So of course, every segment carry different information for making different proteins. Option B. The virus particles contain a virus encoded enzyme that can copy the genome into its complement. When the virus has RNA, it must have an enzyme to do further processing with that RNA. Option C. Purified RNA extracted from the virus particle is infectious. We discussed it that a purified piece of genetic material or genome is not able to cause disease. It should have its other particles. The option D. The virus can acquire new antigen via resortment of its RNA segment. Now, what is resortment? When two different viruses attack a single cell, they mix their genetic material and produce a new virus. They do so by mixing their genes, and this is called resortment. Now, what our option says, the virus can acquire new antigen via resortment of its RNA segment. Yes, it can make new antigen. So the answer is option C, which is incorrect. Question 162. Latency is an outcome particularly characteristics of which one of the following virus group? What is latency? Latency is like a sleep mode of certain viruses. After infecting a host, instead of immediately causing disease, the virus hide inside the host cell and stay inactive for a while. A classic example of latency is herpes simplex virus. After the initial infection, which might cause cold sores or fever, blister, the virus goes into the latent phase inside the nerve cells. It can reactivate later in life, leading to reoccurrence of the sores. That was the concept of latency. Now, what are the options? Option A, poliovirus. Option B, herpes virus. C, rhinovirus. D, influenza virus. And the correct answer is herpes virus. You have to remember this. Question number 163. Each of the following statements concerning viral serotype is correct except. What is serotype? Let's see what the book says about this. We will turn to page number 217 from our book. The term serotype is used to describe a subcategory of a virus based on its surface antigen. For example, measles virus has one serotype, poliovirus have 3 serotypes, and rhinovirus have over 100 serotypes. Now we know serotype is the subtype of the virus. So in the statement, they want to check our knowledge about viral serotypes. Option A, in naked nucleocapsid viruses, the serotype is usually determined by outer capsid proteins. What is naked nucleocapsid viruses? I told you the concept of envelope. If the virus is enclosed in an envelope, it is called enveloped virus, and if it is not, it is called naked virus. So in naked virus, the outer coat is protein, and we studied it before that when the virus makes some changes, it produces antigen on its surface. So in naked viruses, the surface are protein coat. Of course, they make changes on that coat. So this statement that the serotype is usually determined by outer capsid protein in naked nucleocapsid viruses is correct. Sorry, I forgot about explaining capsid. Let's turn page number 216. It says the nucleic acid is surrounded by a protein coat called capsid. I think option A is cleared. And option B, in enveloped virus, the serotype is usually determined by the outer envelope protein, especially the spike protein. If the outermost surface is enveloped, it shows some changes on envelope, and these changes are spike proteins, which tell us about its serotypes. Option C, some viruses have multiple serotypes. Yes, we know some viruses have multiple serotypes. Option D, some viruses have a RNA polymerase that determines the serotype. Yes, it is true that changes in the genomes create different serotypes, but we recognize them by their surface, not by RNA polymerase. So this option is incorrect and fulfill the criteria of our question. Question number 164. The ability of a virus to produce disease can result from a variety of mechanisms. Which of the following mechanism is least likely? To solve this MCQ, we should have the knowledge of virus pathogenicity, that how virus produce disease. Let's turn page number 243 from the book. There are 4 main effects of viruses infection on the cell. Number 1, death, which is also called cytopathic. Number 2, fusion of the cell to form multinucleated cell. Number 3, malignant transformation. And number 4, no apparent morphological and functional changes. Coming back to the question, let's read options. Option A, cytopathic effect in the infected cell. Option B, malignant transformation of infected cell. Option C, immune response to virus-induced antigen on the surface of infected cell. Option D, production of an exotoxin that activates adenylate cyclase. Do I need to tell you the answer? Yes, you are right. Answer is option D. I don't want you to get bored. So take a break, grab a cup of coffee, and see you in next video. And if you like anything in my video, then please hit the like button and subscribe to my channel.